Good afternoon. Today we're discussing Vibrio cholerae, the bacterium causing cholera. This presentation covers its key aspects, impact, and control. Introduction and description. Vibrio cholerae is a gram-negative, comma-shaped bacterium responsible for cholera, a major global health concern, especially where sanitation and clean water are scarce. Understanding it is vital for careers in healthcare, microbiology and biotechnology, environmental science and engineering, and even international development and humanitarian aid. Despite its harm, studying V. cholerae has significantly advanced public health, driven scientific research, and highlighted global health disparities. Vibrio cholerae is a prokaryotic, gram-negative, comma-shaped bacillus lacking a nucleus and organelles. The key components include the cytoplasm, a single circular chromosome, ribosomes for protein synthesis, and the cell membrane, which controls substance movement. Other features, like the flagellum, pili, and capsule, are also present. The cell envelope is gram-negative, featuring an outer membrane with LPS endotoxin, a thin peptoglycan layer, and a periplasmic space. External appendages include a flagellum for motility, pili for host cell adhesion, and a glycocalyx for adhesion and protection. Internal structures include plasmids and inclusions/granules, which is nutrient storage. These are vital for its survival and disease-causing ability. Vibrio cholerae multiplies rapidly through binary fission, where one cell splits into two identical cells. For study, scientists culture them in labs using various media, enriched for optimal growth, selective media to isolate, and differential media for visual distinction. As a heterotroph, Vibrio cholerae consumes organic compounds for energy. Its environmental preferences are key. 1. It's considered a mesophile, which lives around moderate temps 20 to 40 degrees Celsius. It is also considered an alkalinophile, which is a pH of 7. Facultative, it is also a facultative anaerobe that lives with or without oxygen, and is also a halophile that loves living in salty environments. Understanding these help control its spread. Their workings involve metabolism, which breaks down compounds for ATP. As facultative anaerobes, they adapt to varying oxygen levels, with enzymes accelerating reactions. Their genetic blueprint is DNA, typically two circular chromosomes in a cytoplasm's nucleoid region, without a nucleus. Vibrio cholerae adapts through horizontal gene transfer, which considers conjugation, transformation, transduction, and also mutations in its DNA. These changes can alter traits like antibiotic resistance, virulence, and environmental adaptation, posing ongoing public health challenges. Controlling Vibrio cholerae is vital. Preventing transmission through contaminated water or food is central via sanitation and disinfection. Physical methods for water treatment include boiling, filtration, and UV light. Chemical methods use halogens, for example, chlorine and iodine. For water and alcohols for surfaces, these environmental controls are vital for preventing outbreaks. Beyond environmental controls, medical and public health strategies are vital. Antimicrobial therapy uses antibiotics through antibiotic resistance is a major concern. Prophylaxis and combined therapy are also employed. Public health and epidemiology involves studying disease patterns, identifying reservoirs, and conducting surveillance. Vaccination is a key preventative tool. Effective control requires a combined approach of sanitation, water purification, medical treatment, and public health efforts. To detect a Vibrio cholerae, we first get the sample. Usually comes through a stool, poop. and inoculated onto selected media like TCBS, agar, then incubate. Phenotypic methods include microscopy, comma-shaped motile gram-negative, and even colony appearance, which example is yellow on TCBS agar. And lastly, simple chemical test, which is a biochemical reactions for confirmation. More advanced detection uses genotypic methods to find DNA fingerprints. PCR amplifies DNA through toxin genes. DNA probes bind to sequences, and whole genome sequencing provides detailed genetic information. Immunologic methods use the body's defenses. Agglutination test clump bacteria with antibodies. ELISA detects antigens, antibodies, and rapid strip test, RDTs, offer quick point of care results. Combining methods ensures accurate identification. Vibrio cholerae enters orally, survives stomach acid, and colonizes the small intestine, unfortunately. It produces cholera toxin, CT, causing severe watery diarrhea, massive fluid loss, dehydration, and potential death. The slide also reiterates immunologic detection methods from slide 12. Agglutination test, ELISA, and rapid strip test are crucial for accurate diagnosis. Beyond initial defenses, adaptive immunity activates antibody production by B cells, especially secretory IgA, or if you want to call it SIgA genes, neutralizes toxin and prevents attachment. T helper cells coordinate the response. Immunity and vaccination. Natural infection leads to temporary acquired immunity. Vaccination includes a protective immunity without causing disease. Understanding this interaction is crucial for effective treatments and prevention. To summarize, who is Vibrio cholerae? A tiny comma-shaped gram-negative prokaryote causing cholerae, motile, and adhesive. Next, why is it important? It's a global threat with high career relevance in healthcare, science, environment, and humanitarian aid. And its study offers humanities lessons, advancing public health and highlighting global inequalities. Vibrio cholerae multiplies rapidly through binary fission, grown in labs on selected differential media. It's a heterotroph, preferring moderate temperatures, alkaline and salty environments, and it's a facultative anaerobe. It adapts through horizontal gene transfer, through conjugation, transformation, transduction, and it goes to mutations in its DNA, leading to antibiotic resistance and enhanced virulence. Finally, Vibrio cholerae causes sickness by entering orally, releasing cholera toxin and causing severe dehydration. Our immune response includes innate defenses and adaptive immunity, antibodies. Natural infection offers temporary protection, and vaccines teach the body to fight. Detection uses lab methods, PCR, and rapid antibody tests. Treatment focuses on rehydration and antibiotics. Control hinges on clean water, sanitation, and vaccination. Overall, I chose this disease because I have had a similar disease, definitely not pertaining to any kind of death. However, I did have the 24-hour bug, which caused me to throw up continuously for 24 hours. And that's very similar because this Vibrio cholerae affects the intestine, but it's worse than a 24-hour bug. It could cause literally death. But do your best to be careful what you drink, what you touch, what you sniff, and even what you eat. and make sure everything's fully cooked, and make sure things are clean. Um, Thank you for your attention, and I'm happy to answer any questions. These are the citations. Goodbye.